Good morning. My name is J.K. Devine. I'm with the University Relations Department at the University of North Georgia. This morning we are here today with Dr. Ashley McCaskill, who is going to tell us all about our Titum arum plant, Carol here. We are going live to you to allow you to ask as many questions as you would like of Dr. McCaskill, who has tended to Carol for the past 8 years, and just on Friday she bloomed. So we wanted to share this wonderful experience with you. So if you will, Dr. McCaskill, can you please tell us a little bit about Carol?
Yes, so Carol's scientific name is Amorphophallus titanum. She belongs to the Araceae family. She is actually native to Sumatra, so this is definitely not, excuse me, her home territory. She has several common names, including the Titan arum, but also the corpse plant. And she gets the name corpse plant because of the extremely foul odor she emits when she's in her blooming state like she is now. And she has been smelling since Friday. The odor's not quite as strong today, but it's still pretty unpleasant. We've had Carol, like JK said, for about 8 years, and this is the very first time she's bloomed, so we are just ecstatic. Now, I had been planning on having a big party so everyone could actually come visit her in person when she bloomed, but unfortunately, in the current circumstances, that wasn't possible. But thank goodness University Relations set this up so we could do a live stream and people could at least get to see her, even if it's not in the flesh, no pun intended. So the structure that you see here is actually not one flower. This is what we call an inflorescence, and it's a collection of lots of flowers that are hidden down inside. So the morphology is typical for the aroids, which are the plants in the Araceae family, and it's composed of this outer spathe here, and then this internal spadix or column. Now the flowers are at the base of the spadix. Now you're probably wondering why it smells so bad, and descriptors I can give are of rotting meat, or my personal interpretation was the pit toilets at a state park. That's what she smelled like to me. You might wonder why a plant would smell so foul. Well, it's to attract pollinators. And in the wild, Carol's native pollinators would be carrion beetles and flesh flies. That's what is hypothesized. And they think they're going to get a meal because they feed on rotting flesh, but instead they just end up picking up pollen from Carol or whatever Titan arum they're visiting, and then hopefully carrying it to another Titan arum for pollination. Underground, Carol has a structure called a corm, and here I have a corm from another species of arum. This is Amorphophallus bulbifer. It's one of Carol's cousins, but it's much smaller. And this structure that looks like a ball of dirt is actually what we call the corm. It's a modified stem that stays underground, and it's designed to store lots and lots of carbohydrates. So she has a corm in her pot that's probably about this big, and they can get up to 70 pounds, I believe. Well, extremely large. You can see the little roots coming off the top here. And this little bulbifer here is about to send up a new leaf. You can see that's the shoot right there for the new leaf. Back here, we actually have another Amorphophallus titanum. We're fortunate enough to actually have 5 of these in our collection. And this one is currently in its vegetative state. This thing that looks like a tree is actually one single leaf. One leaf. Isn't that amazing? Carol's last leaf was even bigger than this. I believe it reached close to the top of the rafters here in the greenhouse. And so these Amorphophallus plants will go through a period of dormancy. So when this leaf is done doing all of its photosynthesis, it will actually die back and go dormant for a period of time. And then after a few months, it will send up a new leaf. Or, if we're fortunate, an inflorescence like Carol. Here is one of our other Amorphophallus. It's currently in the dormant state. This is Stephen, and Stephen is about the same size as Carol, so fingers crossed Stephen will bloom for us in the next year or two, and we can actually have people come into the greenhouse and get to see it in real life.
So we have our first few questions. We know you've already answered them, but just to give a shout out to the people who are watching, Deidre Burrows wonders, what does it smell like?
Yeah. People have different impressions, but it's usually something pretty awful is what they describe it as. In fact, one person that was here the other day, the odor was so strong and foul, she got very nauseous and had to leave.
And Melanie says, what is the typical root size and expansion for this plant?
Oh, typical root size. So the corm itself, which isn't a root, it's a modified stem, like I said, can get up to 70 pounds or I think it's pounds. Don't quote me on that. But extremely large, but that takes many years to reach that size. In fact, when Carol goes dormant, we're going to have to repot her and transplant her to a larger pot.
Well, apparently one of your biggest fans, Valerie Rest Fambro, wants to know, how does Carol make that smell? And then she comments, is it because a skunk puts its stink on it, or does Carol have her own smell to her?
Carol actually is producing these compounds, and there's several compounds that are being produced and emitted that will attract the pollinators. In addition, the spadix itself actually heats up. It generates heat, which normally you don't associate with plants. But the heat that it produces is to help disperse that odor and send it out further distances to attract even more pollinators.
All right. A watcher, Joy Bolt, wants to know, how old is Carol and why was she named Carol?
So Carol is probably about 10 years old. I don't know how old she was when I got her, but we've had her for about 8 or 9 years. Carol is named, we were just so excited here in the biology department, and we were trying to figure out a name, and somebody just popped off Carol. And it seemed appropriate to me because, well, when I was in 2nd grade, I had a teacher named Mrs. Henry, and she had a guitar that she would play for us, and she named it Carol so that it could be applied to a girl or a boy.
Your favorite student, once again, wants to know, how that is it just one leaf over there, or why isn't it the shape of a regular leaf?
Well, leaves actually come in all sorts of shapes and varieties. If you think about it, let's see if we've got some good examples. Well, think about an aloe vera. The squishy leaves there, those are a different type of leaf. Leaves are actually the most variable of all the plant organs. So there's a huge diversity of actual leaf shapes and sizes, and some plants don't even make leaves.
And we have plenty more questions coming in. Amanda Joy Rudder wants to know, how is such a big plant stable in such a relatively small pot?
That's a good question. I was worried that she might be too top heavy, but her corm is so large that it gives her a good solid base there. And this is actually hollow. You probably can't hear that, but it's actually not as heavy as it looks.
Gloria Patton Roy wants to know, how much does Carol cost?
I believe when we ordered her, she was about $100, which in my opinion was a bargain because I was so excited to get one of these plants.
And for me, how rare is this plant?
Well, it's only found on the island of Sumatra, that in itself makes it pretty rare. But unfortunately, due to habitat destruction, it's even becoming more rare, and now it's actually considered endangered. Although there are quite a few specimens in cultivation in greenhouses and conservatories and botanical gardens all around the world.
And where were you able to get Carol? And that is a question from Mary Swanson.
I got Carol from a nursery, a mail order nursery called Plant Delights Nursery. They have a wonderful selection of many different species of Amorphophallus, as well as other very interesting plants.
Julie Montgomery would like to know, how often will it bloom, and how long does somebody like Carol here, or excuse me, some plant like Carol here live?
So the bloom is very unpredictable. I was not expecting her to bloom this time. I thought it would still be several more years. One of my colleagues said that her former institution, they had one that would bloom every 2 years, like clockwork. But in other circumstances, it can be very unpredictable. So it may be several years before she blooms again. As far as how long they will live, that is an excellent question that I don't have an answer for. Plants are interesting in that many times, if they're in the right conditions, they can live theoretically indefinitely. The oldest plant is actually several thousand years old. It's a bristlecone pine in California, and it's estimated to be, I think, almost 5,000 years old.
And Mary Elise Cox wants to know how many of these plants are there in the world?
Oh, that's another excellent question that I can't answer. I don't know that there's actually a population count for those in the wild or in cultivation, especially now since they're available commercially. Their numbers are increasing in cultivation.
And Heather York Wisely, and I apologize if I mispronounced your name, wants to know, how does Carol get her colors?
Oh, plants make all sorts of wonderful pigments for attracting pollinators or seed dispersers. I am not sure exactly what pigment is being produced here, but my guess, based on the shade, is that it's some sort of anthocyanin, which are purple, red, pink pigments that plants make.
Mitney, and I will butcher your last name, so I'm not going to use it, says, what does the plant look like in her dormant state?
Not much. So in the dormant state, it's just going to be the corm, which we can't see because it's buried in the pot. But pretty much something like this, relatively unimpressive brown ball.
Joni Chimbers wants to know if we can see the flowers down inside Carol. Joni, I'm going to answer that for you. If you wait closer to the end of our hour, Dr. McCaskill here is actually going to...
We're going to excise the spathe. I've never done this since this is our first bloom, so we're all going to be on this big adventure together. But my plan is to actually cut off the spathe here so that we can actually see the flowers inside. And I hopefully will collect some pollen as well.
So please stick around for the full hour. It's going to be great. Please keep your questions coming on in, but I do have a question for Dr. McCaskill right now. Can you tell us what exactly are the different parts of this plant?
Yeah, so in the florals, when it's in its reproductive phase, this type of plant has some very specific parts. Down here we can see some bracts that fell away. They were covering the bud while it was developing. Then this outer sheath here is called the spathe, and it encircles the spadix here. The spadix contains the flowers down at the base, and the spadix is also what generates and disperses the odor.
And tell me, how does Carol reproduce?
Well, normally in the wild, Carol would be pollinated by carrion beetles or flesh flies coming in. They are attracted to the foul odor, and they'll pick up the pollen and then disperse it to another Titan arum. Now the flowers, Carol actually has both male and female flowers down inside her spathe, and they actually develop at different times. The female flowers will develop first, and then later the male flowers will develop. And the goal there is to promote outcrossing with other Titan arums and maintain hybrid vigor, or not self-pollinate is the goal here, to increase the genetic diversity.
Now you mentioned that you're hoping to get some pollen from Carol. How would you go about getting that?
Well, I brought some tools. I have some paper and a paintbrush that I'm hopefully going to just swipe off the pollen. It actually gets extruded out of the male flowers in these little strands, from what I understand.
And what made you decide to get this particular kind of plant for UNG's greenhouse?
Well, I wanted to fill the greenhouse with some very charismatic, interesting plants to pique my students' curiosity. Many times, when they think of plants, they just think of trees and grass. And I wanted to really show them that there are some amazing things out there in Kingdom Planty.
And how many other Titan arum plants do we have that are like Carol, for people who are just joining us?
We actually have 5 of these specimens. I was fortunate enough when I was repotting Carol the first time to discover that we had multiple corms inside the pot, so I separated them out, and so now we have 5 different plants, individual plants at different stages of growth. 2 of them are relatively small. Stephen is about the same size as Carol, leaf-wise, and then this is our middle child, mid-sized.
And what makes this plant so rare and unique?
Well, obviously what makes it unique is the horrible smell it makes, although there are quite a few others that also smell really bad. But also this makes the largest inflorescence on the planet. So it's one of those record breakers in the plant kingdom up there with the giant redwoods of California. The inflorescence can reach a height of up to 8 feet when they are older. Carol's inflorescence here is relatively small, probably about 5 feet, because this is her first bloom. But the next time she blooms, it will probably be much larger.
All right, we want to thank you for keeping your questions coming in, so I'm going to go through some of them with Dr. McCaskill. Charlotte wants to know how, because you mentioned the bloom, she wants to know how does the flower bloom and open up?
Well, it starts out sending up a little shoot, and we'll go back to this little guy, except of course it's going to be much, much larger in Carol's case, but a little shoot comes up out of the ground, and it's covered by the bracts here. And it will actually just continue to get bigger, and the bracts will fall away, and then you'll start to see the spathe appear, and then the spadix will start to emerge from inside the spathe. And then eventually the spathe, when she was fully open 2 nights ago, was expanded out.
And speaking of how, if you're curious about how she expands out, we will have a time-lapse video on our website following this Facebook Live video in a couple of hours later this afternoon, so you yourself can see how Carol here opened up and bloomed for us. But in the meantime, keep your questions coming. Troy Callahan asked, how is it able to generate heat? How many pollinators does it need to attract for successful pollination? And please describe the changes the plant will undergo after pollination.
Okay, let's see if I can remember all those questions.
We'll start with the first one again. How is it able to generate heat?
There are chemical reactions that occur in the cells of the spadix that generate and release heat.
And how many pollinators does it need to attract for successful pollination?
That's another very good question I can't answer, but obviously the more pollinators you attract, the more successful you're going to be at getting pollinated.
So what are Carol's normal pollinators in the nature?
So they are the carrion beetles and the flesh flies. That's the hypothesized pollinators. In fact, I think it was only within the last decade that they actually were able to confirm that those were her actual pollinators.
Please describe the changes the plant will undergo after pollination.
So what will happen is the spathe will eventually wither and fall away. It will senesce, that's a botanical term for dying. And if she was successfully pollinated, we would then see fruit develop at the base of the spadix here, and the fruit develop into red berries, bright red berries. And in Carol's case with an Amorphophallus titanum, they're probably, I haven't seen them in real life, so I'm kind of guessing here, but probably the size of an olive. And in the wild, the fruit are actually dispersed by hornbills and other birds, or the seeds are dispersed, excuse me.
And once they're pollinated, again, our favorite question, how long does it take to bloom?
Well, once it has to bloom again after this goes away, it's probably going to be several years before she blooms again.
Valerie wants to know, how long does it stay in its dormant state phase, Dr. McCaskill? She specifically said Dr. McCaskill.
So it's variable. It's usually several months that they will remain inactive and dormant.
And Gloria Patton-Roy has a question, how long did it take for her to flower?
It actually happened very quickly. When the shoot started to come up, I thought it was just another leaf. But then we got the wonderful surprise that it was actually an inflorescence coming up. And that was mid-March or late March, I believe. And so within a couple of weeks, she grew. She was growing at a rate of 3 inches a day there for a while.
Okay, we still have a few more questions coming in. Again, please keep them coming. Valerie says 6-year-old Charlotte wants to know, do birds that eat dead stuff try to eat it?
Oh, good question, Charlotte. No, not that I'm aware of, but she's not outside where birds could get to her. So,
Joy Bolt wants to know if Tell me the middle child is named Jan.
Since this one doesn't have a name yet, we might have to have a contest to come up with a name for the middle child.
That is a great idea. So if you don't have a question, but you have an idea, we have Carol here, and what is our other child's name?
Stephen.
We have Carol and Stephen. So if you have a suggestion for our middle child, please go on Facebook Live and post your names, suggestions. And we still have a few more questions coming in. Somebody who is joining us just now, please let us know. We are at the UNG Dahlonega campus in their greenhouse, and this is where Carol has been growing for the past 8 or 9 years, and she is blooming. Okay, and someone asked us to clarify the question about whether a skunk had rubbed on it from Valerie's other 6-year-old daughter. So, no.
No skunks were involved in the stink.
And tell us again, what does the stink smell like?
It smells, to me, like a bathroom in a state park, one of the pit toilets. Other people say it smells like a rotting carcass, which hence the name corpse flower. But it's very, very unpleasant, and it sticks with you, as it turns out. My clothes, when I got home after being with her the first night she bloomed, smelled like Carol. And my purse still smells like Carol. I don't know how I'm going to get rid of the odor on my purse.
Well, we do know from one of our other technicians who is here on hand, says Febreze seems to work well to expel the heat. But back to a few more questions. Tom and Julie Long want to know, what do we use for fertilizer for Carol?
In this greenhouse, we actually just use Miracle Grow. We keep it pretty simple.
And tell us, what other things do you have, or excuse me, what other plants do you have in this greenhouse besides Carol?
We have other species of Amorphophallus. We have the Amorphophallus bulbifer and an Amorphophallus konjac. We also have carnivorous plants, Nepenthes. We have a diversity of ferns. We have quite a few cacti. And then my other big goal for the greenhouse, besides the exotics, is to have a good diversity of food plants. So I have a vanilla orchid and a coffee plant and mango tree and an avocado tree, so that students can actually see where their food comes from. Oh, a black pepper plant, that's when I'm excited to have it bloom.
Right. Mary Swanson asked, have any other plants in the greenhouse reacted to Carol's terrible smell?
Not that I'm aware of. Plants are pretty immune to what's going on around them.
We have a question from a 5-year-old Emmeline. She wants to know about the plant. So I didn't quite see her question, but why don't you give us one more overview of Carol for those who are just joining us. Please keep your questions coming, but I'll let Dr. McCaskill go once again, give us an explanation about Carol.
Yeah, so Carol is an Amorphophallus titanum. She belongs to the Araceae family. She is native to Sumatra. That's the only place in the world you can see her in the wild. And unfortunately, due to habitat destruction, she's now considered, or the species is now considered endangered. The structure that we have here is an inflorescence, so it's actually a collection of flowers. This isn't one flower. All the flowers are actually buried down inside this outer leaf-like structure that we call a spathe, and the spadix here is the central column. So the function of the spadix is to generate and disperse the odor that attracts the pollinators. And then underground we have a modified stem called a corm that looks something like this, except Carol's corm will be much, much larger, probably about the diameter of the pot that she's in.
Can you tell us about the colors of Carol and what she smells like, and tell us about her kin in the greenhouse?
So the colors are part of the mimicry. So in addition to the odor that attracts the pollinators, the color is supposed to also mimic flesh, rotting flesh, basically. It's most likely anthocyanins, but I haven't done the reading to determine exactly what the pigment is. But anthocyanins are common flower pigments. What was?
And who are her related people here in the greenhouse?
So we also have, this is an Amorphophallus bulbifer. And this is about as big as the leaf gets in this species. You can see there's a new leaf coming up right here. The Amorphophallus bulbifer also stinks. I had one of these in my office. I took it to my office so I could show it to my students. And I left it there overnight and it opened. And the next day I could smell it when I got off the elevator. In fact, other faculty called facilities because they thought there was a dead rat in the building. It smelled that bad. So now I have a bad reputation in the building for having stinky plants around.
And who are some of the other Titum arum plants do we have here?
Okay, so other Titan arums, we have this one that hopefully will get a name one day soon. That's vegetative right now. We have Stephen right here that's going through his dormant phase. Stephen is actually about the same size as Carol, so I'm hoping that Stephen will actually bloom in the next couple of years, fingers crossed.
And for those of who are just joining, how are you able to come to acquire Carol?
So I was fortunate enough to discover that Plant Delights Nursery, they have a catalog and website. They're a mail order nursery, was actually selling them. I didn't know that you could actually buy them, so I was ecstatic when I found that out.
And why did you want to buy Carol?
I wanted Carol because of this. It's so impressive, and I wanted to have some very charismatic plants in the greenhouse that would really draw in my students and show them how amazing plants actually are.
And where does Carol come from originally?
So where is she native? She's native to the island of Sumatra, and I believe that's the only place on the planet where you can see these in the wild. That's part of Indonesia. Interestingly, there's another amazing plant in Sumatra called Rafflesia arnoldii that's also called corpse flower for obvious reasons because it stinks, but it actually makes the largest single flower on the planet. That the flower can reach 1 meter in diameter. But unfortunately, it's a parasitic plant, so it has not been cultivated. We haven't been able to cultivate that. So you have to actually go to Sumatra to see the Rafflesia.
And how are you able to cultivate and grow Carol?
Well, Carol, because she's a corm, they shipped me the corm, and I just put it in a special blend with lots of peat and soil, and then just treat her like any other plant. Water her when she's thirsty, feed her on a regular basis.
And how tall does Carol get?
Well, Carol, the inflorescence can reach up to 8 feet. So every time she blooms, the inflorescence will most likely be larger until she reaches that max height of 8 feet. The leaf, though, can reach a height of 10 meters. So this leaf right here is relatively small. Carol's last leaf was much, much larger and actually reached the rafters here in the greenhouse.
And how long does it take somebody like a Titum arum like Carol to bloom the first time, and then how often does she rebloom?
So the blooming is actually relatively unpredictable. I was not expecting this. I thought it would be several more years. Usually they say it takes about 10 years for them to bloom for the first time, because doing this requires a lot of energy. So they have to photosynthesize for many years to store up enough energy to be able to do this. Now, the bloom cycle is, again, unpredictable, unfortunately, but it will probably be a few years before she blooms again.
And how long, once Carol blooms, does the actual bloom last?
That is another thing that makes her really special, is that the bloom is actually short-lived. She opened Friday night, and then you can see what she looks like now. So it really only was fully expanded and at its peak for about a day.
All right, we're still going to be continuing answering your questions for about 15 more minutes. So if you have some, please send them in. But after that, we're going to watch Dr. McCaskill here kind of, I guess, dissect a little.
Yeah, we're going to do a little Amorphophallus dissection.
So Katie Hinman wants to know, in the wild, do these bloom seasonally?
Well, since they grow in the tropics, they're not going to be under that seasonal cycle. It's probably more of a wet-dry cycle, I would guess.
And also, as we mentioned, if you are just coming in with us, we have 2 names for our Titum arums here. We have Carol and Stephen in his dormant state, and we have one in our vegetative state. So if you have a suggestion, please send them in, because 6-year-old Charlotte wants us to name her Cherylanda.
Cherylanda.
Cherylanda. All right. And Carrie Leon asks, what is the typical routine for taking care of Carol? And how often do you water her and the others?
Well, it's going to vary as far as watering goes, depending on what the temperature in the greenhouse is. In summer, she gets watered more frequently because it's much, much warmer in here. In the winter, probably not as often, a few times a week. And the plants in the greenhouse generally get fertilized about once a week.
Speaking of the greenhouse, we've had actually a lot of questions about that. Lauren Bradshaw wants to know, when we are not social distancing, are people allowed to visit the greenhouse?
Well, it stays locked at all times for obvious reasons, but I'm always more than happy to have visitors come. They just need to make arrangements.
Well, Sherry Brown Griffith asked, during family weekend, would we be able to see Carol? Or will she, when COVID is over, or will she not be in this state by the time we have family weekend next fall?
Unfortunately, she won't be in this state any longer. We'll probably have leaves, but even though they are impressive, they're not quite the same as this. But the next time we have one bloom, assuming that we are no longer socially distancing, I plan on having a really big public event where people can actually come and see and smell for themselves.
Deanna Magoon, I hope I pronounced that right. Again, I apologize if I'm butchering anyone's names. What type of classes do you teach here at UNG?
Well, I am a plant biologist, so I teach general botany and plant physiology and plant taxonomy and botany field study. And then I also teach mycology, which is the study of fungi.
And you are actual botanist, correct?
Correct.
All right. Natalie Macy wants to know, how big was Carol when you got her? And she specifically says her mom would like to know.
So Carol, if I remember correctly, the corn was probably about this size. That was so long ago. I've slept since then. So.
All right. Will Mann asks, do you know about the corpse plant that was growing in Athens in 1840 in the first university botanical garden?
No, I did not.
Well,
I love learning new trivia.
Well, thanks, Will. Marianne Johnson wants to know, where did we get the plant?
So I ordered her from a nursery called Plant Delights Nursery. They are out of North Carolina, I believe. And they actually have quite a few species of Amorphophallus. So if you don't have space for a plant like Carol, you can get other species like the Amorphophallus bulbifer, which maintain a much more reasonable size.
Leanne Hicks would like to know, is Carol self-pollinating?
Well, typically no, because of the staggered development of the male and female flowers. That is going to promote outcrossing. But I am going to try my hand at some pollinating today or tomorrow. And we'll see if we can get some self-pollination happening.
Sherilyn Rumley wants to know, how long does it take for a leaf to grow?
The leaf, I haven't actually timed it, but it's probably a month or two to reach full size. And then they'll last for months.
Well, please keep your questions coming on our Facebook Live, and if you want to see Carol blooming, we will have a time-lapse video available on our Facebook page as well as all of our other social media platforms. And we'll be here for probably about a little less than 30 more minutes, but in about 15 minutes, Dr. McCaskill here, we're going to open up Carol and see what those flowers are inside. But right now, Dr. McCaskill, for those of us who are just joining us, can you please once again tell us a little bit about Carol, how you acquired her, and what you did to get her to this wonderful state?
Yes, so Carol is an Amorphophallus titanum, that's the Latin name, and she's a member of the Araceae family. Other plants in this family that you may know are calla lilies and peace lilies. Now, and those aren't actually lilies, though. It's one of the problems with common names. Carol's common names are the Titan arum and then also the corpse plant. And it gets the common name corpse plant because of the very, very offensive odor that is produced when it's blooming. It smells like pit toilets at a park, rotting carcass. And the idea there is that that odor attracts pollinators, specifically carrion beetles and flesh flies that normally feed on rotting meat. The structure that we see here is actually an inflorescence. So this isn't one flower. And there's actually hundreds of very small flowers down inside the spathe. So this structure is the spathe, and this is the spadix. And this morphology is typical for the Araceae family. A species that's native to here that you might be familiar with is jack-in-the-pulpit, Aracema triphyllum. The pulpit is the spathe, and the jack inside is the spadix. So the spadix does a couple of things. It actually produces the foul stench, and then it also heats up. And the heat is there to help disperse the odor even in greater distances to attract even more pollinators. Carol is native, her species is native to the island of Sumatra in Indonesia. And unfortunately, due to habitat destruction, Amorphophallus titanum is now considered an endangered plant. Luckily, there are quite a few specimens in cultivation all around the world, so we're not necessarily in danger of totally losing the plant, but we may not see it in the wild for much longer unless things improve. The color here inside the spathe is also part of the mimicry that is going to help attract seed or the pollinators. So it's dark red maroon like rotting flesh would be. Underground, Carol has a structure that looks something like this. This is what we call a corm, which is a modified stem, and it actually stays underground, and its function is to store lots and lots of energy. Carol's corm, though, is probably about this size. It's extremely large. We've had Carol here about 8 or 9 years, and this is the very first time she's bloomed. So we are just ecstatic and so happy that we can share her with people, even if it's not in real life.
All right, Dr. McCaskill, we have a few more questions still coming in. Johnny Aganovich his 4-year-old son wants to know how the bloom opens and how does it move?
So the bloom, or the spathe, actually expands and unfurls. And there's going to be, this is what we'd call a nastic movement or a turgor movement because it's only temporary. And you can see here that it's actually closed up. So cells are enlarging and then shrinking, and that's what allows it to open up and then close back up.
And Minty would like to know, how long will she stay in bloom?
Well, she's on her way out. These are very short-lived inflorescence. They're fully open for like 24 to 36 hours, and she will now begin to senesce. Now, if we manage to successfully pollinate her, the spadix will remain intact to allow fruit development, but the spathe will senesce, which is a botanical term for dying.
And Frank asks the question, please explain the relationship to jack-in-the-pulpits if you haven't already done so.
So jack-in-the-pulpits are a native species in the Araceae family, so you can think of them as one of Carol's distant cousins. They also make this similar morphology, but on a much smaller scale. And you have the outer spathe, which actually comes up and folds over the spadix in the interior.
And Mary Ann Johnson has another question. How long does the smell last?
It's really strong and pungent for the time that it's open and fully expanded, so the 24 to 36 hours, but it still smells. But at this point, you have to kind of stick your nose in there to get the odor, but it's still ripe.
And I'm just going through a few more questions. One person asked, Courtney Cordova, what type of environments do they thrive in best?
They are native to tropical forests, so they need year-round warm conditions. This plant would not survive outside here in North Georgia. So it has to be grown inside in a greenhouse, ideally where it's nice and warm all the time, or at least indoors.
All right. And one person asked, so sorry, are they toxic?
Oh.
Leanne Hicks, thanks for your question. She asked, are they toxic?
Well, members of this family, of the Araceae family, actually do accumulate a compound called oxalic acid crystals, and so I am not sure about the Amorphophallus bulbifer, but some of the others, if you eat them, they can make you sick, specifically the corms. I was actually working with one of the cousins of Carol here in the greenhouse, dividing the root ball, and the oxalic acid crystals actually caused my hands to start stinging.
Leanne Hicks actually asked, what does the fruit look like?
They're red berries. The size, I've never seen them in real life, so I'm going to guess here, but I'm predicting they'd be about olive size.
And Frank Corto asked, what does the amorphophallus mean?
Amorphophallus. Yeah, so that's Latin referring to the shape of the spadix. Amorpho means misshapen, and then the phallus refers to male reproductive anatomy.
And tell us once more, how did you come by Carol, and a little bit about her history?
Yes, so Carol was purchased from a nursery called Plant Delights Nursery. They're out of North Carolina, and they have many, many species of Amorphophallus for sale on their website. As far as I know, that is one of the only places they're commercially available. So I was ecstatic when I found out that I could buy one from there. So we've had Carol for about 8 or 9 years.
All right, for those of you who are just joining us, we are at UNG, University of North Georgia's Dahlonega campus in their greenhouse here with Dr. Ashley McCaskill, and we are about to start looking inside. So we're going to let Dr. McCaskill get her tools that she needs to actually start kind of opening Carol up and seeing what's inside her. And so, yeah, but as you go, so one person actually asked, can you see this in person? Actually, this is in UNG's greenhouse. You would need to contact the biology department in order to get in. For those of you who are interested, they do allow volunteers to help here in the greenhouse. Is that correct?
We do have a few volunteers. The Georgia Mountain Master Gardeners, a couple of their members volunteer here in the biology department. But mainly we have students that work in the greenhouse that take care of the plants, normally when the university is open.
But right now we are practicing social distancing, which is why we can't let you hear live and in person to see and to smell Carol. So we're trying to do our very best to bring the best of situation that we can give you. So Dr. McCaskill, why don't you first tell us the structure form of Carol and exactly what you're doing while we go ahead, if you guys are all ready. If not, you can still continue asking questions. We will be online until 11. So if you still have some questions, shoot us through Facebook Live, and I will gladly ask them off camera. But right now, we're going to let Dr. McCaskill see what's inside Carol.
Okay. I hope I don't make a mess of this. Never done this before, so we're all going to be learning and experiencing this for the first time together. So the flowers are at the base of the spadix here, and the spathe is this sterile bracteria that's surrounding it. So what I'm going to do, hopefully, is cut off the spathe so that we can expose the flowers. Fingers crossed that this works. So bear with me if I'm going slow. I want to do this gingerly. The texture, in case you're wondering, is somewhat similar to cabbage, I would say.
And what are you using to cut her open?
Just a razor blade. Nothing fancy. Oh, there's definitely pollen in here. Well, there's our spathe right there, if you want to see the inside. Thank you. That's what it looks like inside. Okay, I'm going to have to excise a little bit more here so we can actually see the flowers better.
For those of you who are just joining us, Dr. Ashley McCaskill, a member of the biology department here at the University of North Georgia, is excising Carol, a Titum arum plant, which is also known as the corpse flower. And we are getting a look at her pollen and her flower state. She bloomed on Friday. She was in full bloom, and if you check our Facebook Live page and our social media pages, we will have a time lapse available for you to see how Carol here bloomed. We do have a question from one person. Does the smell get worse now that you have excised Carol?
Well, I can definitely smell her better.
And for those of you who are just joining us, Carol smells like rotting meat, or what was the other visual you want to use?
A pit toilet at a state park. So now we can see the flowers. We have male and female flowers. So the female flowers are these down here, and then the male flowers are actually all of these tiny little things right here. Oh, and you can see the pollen is being shed actively here. So my plan is to collect some pollen, and we'll try and see if we can also pollinate her. I'm not sure if her female flowers are still receptive at this point, unfortunately, but just quite a bit of pollen being produced here. Very exciting. So the flowers actually don't look like what you expect flowers to look like. They are highly reduced and pretty much are composed strictly of the reproductive organs. So we have an ovary with the stigma and style, and then the male flowers are basically just going to be anthers producing the pollen.
Now we do have a question from Risa Newman. She said, did cutting apart Carol's leaf or her flower portion, did that traumatize her or hurt her in any way?
Well, she probably didn't appreciate it, but since that part will be senescing anyway, it'll be okay. She'll be fine. And unfortunately, that's what you have to do to actually get to the flowers. You can see that the spadix is actually swollen here, and so that makes it really difficult to get down in there. Of course, the insects can do it quite easily, but for us, it's difficult.
Yes, Kamari asks, are they both mature right now?
Typically, the female flowers mature a few days before the male flowers to help promote outcrossing. Obviously, the male flowers are mature since they are shedding pollen, but I don't know if the female flowers are receptive any longer. We'll find out. We'll watch Carol for the next several months and see if we have fruit develop. And if we do, that means that she was successfully pollinated.
Right, now how will you go about actually trying to pollinate her?
Well, I will probably just use my finger here to rub off some pollen and then transfer it to the stigmas, which are the sticky tops here on the carpels, and that is where the pollen is designed to land. And once the pollen makes it to the sticky stigma, it will start to grow a pollen tube down inside the ovary to an ovule, which will eventually develop into a seed if the pollination and fertilization are successful.
Holly Easterby wants to know, with you touching it, do your hands smell?
No. Although I did smell yesterday and the day before just from being in the greenhouse with her. But the pollen and the flowers themselves don't smell, it seems.
Valerie asks, can we see some of the pollen on your finger?
Here, let me get some more. I don't know if, can I?
Okay. We tried our best, ladies and gentlemen.
Yeah, so it just looks like yellow powder.
Terry Michelle asks, will the flower continue to live as long as it's taken care of, or does it have a lifespan?
Well, the inflorescence itself has a finite lifespan. It will senesce eventually. Now, if the pollination was successful, then it will live long enough to develop fruit and then have the seeds dispersed by us. And then at that point, the entire structure here will senesce, and the corm will go dormant for a few months, and then it will produce a new leaf that will look something like this, except even larger. And it will produce leaves for several years, excuse me, photosynthesizing and storing up lots of energy so that it can make a flower again in a few years.
Once again, you are here with Dr. Ashley McCaskill at the University of North Georgia's Dahlonega campus. We are in our greenhouse right now, and we are talking about our Titum arum, Carol here, who we have, what was it you called it?
Dissected or excised.
And we are going to be here for about 10 more minutes. So if you have any more questions, please go to our Facebook Live page and post them in the comments, and I will ask Dr. McCaskill. Some of you have asked the question about our time lapse, so some of your wonderful kids at home want to see it. We do have it on our Facebook page. It posted last night, but as soon as we're done here, we will repost it again so you can see how Carol here bloomed. When did she start to bloom?
So she started to open on Friday. Let's see. Well, no, Thursday, excuse me. It's been a whirlwind last few days. So Thursday afternoon, she started to unfurl her spathe, and by Thursday night, she was fully expanded and really, really stinky.
So we will have that for you once again on Facebook right after we end with her, so you can get a good glimpse. It's about 20 seconds, but we'll have a much longer version for you next week from the very beginning when she first started to open till her full bloom. So, and she started blooming later in the evening around dusk, correct?
Yeah, that's typical for the Titan arums. They actually open at night and are at their peak bloom and stinkiness because that's when their pollinators are active.
And for those of you who would like to know how tall Carol is, because that's what interested me, I'm about 6 feet tall, and this is Carol. So she's a little bit shorter than me, but I am standing on rocks. So yeah, that is how big and tall Carol is. We do have a few more questions coming in, though. Johnny asks, what disperses the seeds in nature?
So in the wild, hornbills, which are a type of bird, and other species of birds will eat the fruit and disperse the seeds.
Holly Easterby says, does she produce pups or babies?
So I'm assuming you mean maybe the corm through asexual propagation. Not that I know of.
Valerie asks, is the pollen stringy?
Well, it does appear to be coming out in strands, which is typical for this species. But once you pick up the strands, it's very powdery.
Right. Do you have any more individual Titum arums?
We actually have 5 here in our collection. And there are various sizes. Carol's obviously the largest since she's the first to bloom. Stephen is the second largest, and I'm hoping that he may bloom within the next year. And it would give everyone the opportunity to actually see one of these in real life and smell it in real life.
Marianne Johnson asked, were there signs that she was getting ready to bloom?
No. What you see when they're entering one of their phases, either vegetative or reproductive, is one of these shoots coming up out of the soil. And initially, you can't tell the difference, or at least I can't tell the difference, if it's going to be a leaf or a flower. And it wasn't until the spathe, or excuse me, the bracts started to kind of peel back that we actually saw that was an inflorescence. And I was ecstatic.
Wonderful. Now, for those of you who are just joining us or signing off with us, we are at the University of North Georgia's Dahlonega campus greenhouse with Dr. Ashley McCaskill. Dr. McCaskill, what is it that you do here at UNG?
Well, I'm in the biology department. I'm a professor here, and specifically, I'm the plant biologist on this campus. So I teach all of the plant-related classes and the fungal class.
And you actually started acquiring plants here when our greenhouse here was built about a decade or so ago?
Correct. The greenhouse was built here in about 2010, I believe. And so we started from, with nothing here in the greenhouse. And so over the years, I've been slowly trying to build up our collection of interesting and exotic plants.
Right. And speaking of interesting exotic plants, you picked Carol to bring here. Why?
Because Carol is amazing. And I wanted to have some plants in here that were very charismatic. I know I'm anthropomorphizing here, but really extraordinary and would pique my students' interest. Too frequently, people don't really appreciate plants. They just think about trees and grass. And so if we can have more plants that are really showy in different ways, it helps kind of students appreciate Kingdom Planting a little bit more.
Right, and speaking of piquing interest, we seem to have a lot of interest from a lot of our younger audiences. We have Terry Michelle's 5-year-old who would like to know what it looks like inside of the long yellow part of the plant. So what does it look like inside here?
I'm not sure. I have a feeling that it's hollow inside. That's what it feels like because it's quite squishy.
And Jessica Murray asks, in nature, do they bloom in a synchronized way?
Well, you would think that they would, because in order for pollination between individuals to occur, you would have to have multiple inflorescence blooming at the same time. So there needs to be some synchronicity there.
Right. Dorianne Avis Gilmore says, I have about 10 plants that have come up in my yard in Florida. How can I make sure that I'm giving them what they need? That's a lot of these plants in your yard. But go ahead. How can you assure that she's giving them what they need?
Oh, well, make sure that they, well, of course, it depends on what plants you have, but just make sure that they have water when they need it and appropriate fertilizer occasionally. So that's a hard question to answer because different plants actually have different requirements.
All right. Courtney Cordova asks, referring to the names Carol and Steve, do the plants have sexes? If so, what are the differences and similarities?
Actually, in this species, the plants are bisexual, and they have both male and female flowers on the same plant.
And we do have a few more questions about our time lapse. Once again, we will be posting that once again on our Facebook page and across all of our social media pages after we end here today. If you are just joining us and want to see what we talked about earlier, this will be available on Facebook, and so all you have to do is go back to the actual live portion, click, and you can see everything we've recorded here today. We do have a few more minutes, so please get your last questions in. I think I have one more. Oh yes, obviously. We have Carol here, we have Steve over there, but we are still looking for any more name suggestions for our middle child. Is that correct?
Yes, right here. Send us your suggestions for a good name for this Titan arum right here.
And so you asked which Facebook. It is UNG's Facebook, UNG Facebook page that the time lapse will be on. So, are Carol and Steve the same species?
Yes, they are.
All right. And I think that covers all of the questions that we have had so far. If I've missed any, I do apologize, but you can always comment on them, and we can connect you with Dr. McCaskill, and she can answer those for you, hopefully. Dr. McCaskill, thank you very much. I believe, do we have a few more minutes? Yes, we have 2 more minutes. This is your, as my mother would say, this is your 2-minute warning. So if you have any more questions, please send them in, and we will try to answer them.
And we'll just show off the spathe here that I cut off so you can see for those of you that are just joining.
Ah, yes. Dorianne Avis Gilmore says, can I get a reference material for corpse plants, please?
Reference material. Mm-hmm. So reference material, if you're looking for scientific information, good resources are Chicago Botanical Garden has a good website with lots of information. Also, Kew Gardens in England, they have a good information on their website as well.
And if you have any more questions, please feel free to go to our Facebook page, or you can find Dr. McCaskill on, there is a story about her and Carol on our UNG Facebook page called UNG News. You'll see her standing next to Carol with her leaves. Just click on that and then click on Dr. McCaskill's name. That'll take you to her webpage, her phone number, and her email. So if you have any other questions and you haven't been able to ask them there, I suggest you email her, and she'll be happy to answer them for you.
Of course.
And once again, Dr. McCaskill, thank you so much. But is there anything else you would like to tell the people out there about Carol, or what you would like them to know about UNG and UNG's greenhouse or biology department?
Well, since the focus is Carol today, I just want everyone to know that as soon as I know that we're going to have another bloom, I'll start advertising and letting people know so that you can actually come and see it in person. Seeing it in pictures and on a computer is one thing. Seeing it and smelling it in real life is totally different. So I really want as many people as possible to have the opportunity to see one of these in real life.
Once again, thank you so very much for joining us today on our first Facebook Live at UNG's Dahlonega Greenhouse. We hope you enjoyed this as much as we did.